Hi, thank you very much, Linda. That was fantastic. Really interesting. I just actually one question from me before anybody else puts some. Those three loci, the TP loci, what do they encode? Can you tell us about that at all?
So the locus 136 and 548, they are coding for outer membrane proteins, or they are predicted to code for outer membrane proteins, because in treponema we really have this kind of problem with hypothetical genes, right? So most of the genes are actually predicted in silico. And the 705 is actually penicillin binding protein, one of the penicillin binding proteins.
Okay. Oh, fantastic. And is it a fragment, or it's fragments, isn't it? Not the whole coding sequence that you're...
Yeah, definitely. That's just a fragment. Yeah, that's not the whole one, because when we are using clinical samples, most of the time we are not able to amplify longer fractions. So we were trying to keep it like less than 500.
Yeah.
I mean, that's amazing, because for different reasons. That's what, when we designed MLST in the first place, we kept it so that you could sequence one on both strands, because sequencing capacity was limited. I think it's a wonderful illustration today of how, you know, the practical limitations of trying to do clinical microbiology in an organism that you can't really grow, and it's actually even very difficult to even amplify. You know, that you,
exactly.
Yeah, and I think it's very easy. Everybody gets sort of in this, you know, people say we've sequenced 500,000 COVID genomes, because you know, everybody gets carried away. You can't do everything by genomics. There's sample limitations, and that's one of the reasons why we've stuck with the PubMLST platform, to enable you to have whole genomes and MLST type data side by side, so that you can analyze them together. And it's great to see that, and exactly how you use that sort of multi-locus approach in a slightly different way, because of course Treponema pallidum is as far as we can tell, a very clonal organism with very low diversity. So you can sort of talk about SNPs, but it nonetheless, you do have to have multiple loci, because it's the only way of getting enough resolution. And it's great to see that, because I mean, I don't want to, you know, using sequence-based epidemiology must make this an awful lot easier, because it must be quite difficult to do this by restriction fragment length polymorphism. I know, having spent a long time trying to do that back in the 1980s and 90s before.
Yeah, no, it's definitely difficult, and also the results varies in between labs, right? Because everybody has different eyes, and so it's really old technique, but most of the people are still using that.
Oh, really?
Yes. Because I suppose this is always the problem, isn't it? The first technique that gets adopted, then you know, it's difficult to get people to change. But that's really wonderful. I think it's tremendous work.
Really hard work as well, definitely.
Yeah.
Would those loci be genus-wide, do you think?
Once again, sorry?
Those three loci that you look at, would they be across a Treponema genus? Present across a Treponema genus?
Oh yeah, so actually I'm not sure why are you asking that, but they are across, yeah. Like actually 136 is in the beginning, 548 is in the middle, and 705 in the end. So
I think Azil was meaning, have you got them in different members of the genus Treponema? You do find in other
sorry,
other
no.
species. Not the genome, the genus. That's the problem with Zoom. We could lose something in the.
Yeah. No, it's really like based on Treponema pallidum only. It can distinguish between subspecies, but I'm not even sure if actually different genus or species of Treponems actually have these genes. So it's like based on subspecies and lineages. Yeah.
Okay.
Okay.
Lovely. There's no more questions in the chat at the moment for anyone from anyone. You can ask any. Actually, all well, not 6 but 5 speakers. Perhaps you can switch your cameras on.
I think there was one really good point that Bill made to me in a separate comment, actually, which I think is relevant to all of the speakers, actually. And indeed, Yvonne, who's here, and of course, who spoke at our first event. Good to see you again, Yvonne. Thank you. And Bill was talking about whether you can link specimens from the same patient with different pathogens. And of course, we're not a clinical lab, so we can't do that. But I can quite see that clinical labs might want to do that, where you've got I don't know, you could have indeed all three different organisms present in one patient. But that is, Bill, something that I think we could think about doing, which is linking across the databases, so that, for example, if you had an organism that was a single patient, as long as you have some sort of unique identifier for that specimen that doesn't identify the patient, just says that there's an entry in the Neisseria database, the Chlamydiales database, and possibly even the Treponema database, then we could think about things like that, which might be of value to STI reference labs who are working on all of them together. I don't know. That's just, I thought that was a really good point, and that we haven't started looking at those interactions, because, for example, are some chlamydiales associated with particular gonococci or whatever? Do you get particular, does being infected with syphilis actually make you prone to something else as well? I think there's some quite interesting multi-organism questions which we could think about addressing. We've not done that on PubMST at all yet, but I think it could be done. Keith, are you online? I think something along that line might be possible, mightn't it?
Keith is on the line. He could say something about it. There he is.
Yes, of course, yes, yeah.
So if that was something that the clinical college would be interested in doing, that would be something we could take forward, Bill. I think that was a great idea. Was that what you were sort of thinking of?
Well, a couple of points. As I mentioned, I've always wondered whether anyone has ever found GCDNA in chlamydia and vice versa.
Yeah, exactly.
Yeah. I mean, I don't think that's ever been looked at, but I guess, maybe that's something that could be looked at. But my other point is, I'm just wondering if the technology will advance to the point where we could have a typing scheme based on transcriptomes. And that might give us a bigger insight into the biology of the bug during infection. I mean, at this stage of my career, when I entered into the field in 1979, we were just struggling to do cloning. So I'm just wondering, do you think, Martin, or anybody else, that we could get to the point where we could actually, from a patient sample, develop a typing scheme based on gene expression differences with different types of infections, genital, rectal, pharyngeal?
Well, in principle, the sort of thing that Anastasia was showing, which was looking obviously at the deduced peptide sequences, looking at the peptides. We've always thought, haven't we, Keith, for a long time, that we could build on ways of looking at phenotype information, and transcriptome could be built into that. We've got the indexing of all of the genes, as Anastasia was saying, we got a number. So in principle, that's actually doable. It's just a question of creating the links between the structures and the way of storing that information and linking it to the isolate information. But that is, that's what we would refer to as sort of phenotype information. That's always been a major part of our mission. It's not just cataloging variation, linking, showing trees on maps, important as that is. It's also trying to get to this issue of where the phenotype is. So yeah, I think it's all doable, and it's sort of envisaged within our framework, but it's, obviously, you're sort of talking of almost like a theory of everything. You're trying to do all of microbiology at one go, but that is the ultimate ambition. Has anybody got anything else to add on?
I think that's always something that is certainly linking the genotype with the phenotype would be something that we're quite interested in doing, and equally sort of proteomic data as well. There's just finding the way in which we can do that within the platform. At the moment, we've got genomes, and it's then kind of linking them both together, all of them together in some way. But a transcriptome-based MLST scheme, I'm not sure, how it worked, because it would be across a genome, really, wouldn't it?
But you would start with something like Yolinda's pan-genome scheme, and then you would then actually assign to that a level of expression of each of the genes. for example. So you could, we've done a little bit of stuff like that with some things, but it's very early days.
Yeah, it is.
I think one of the things is when you think about data use and you know, there's lots of, we're not making the most of the data that's available. Lots of people publish all sorts of studies and they put them in various places, usually in PDFs, in journals of variable impact. And then a lot of the data often goes into a PDF in a supplementary table, and it makes it very difficult to link it together. So one of the things we're trying to do is to pull all that together. But it's sort of a big job, and it's a sort of can be easily seen as a bit boring. But you know, I do think that's very much still the way forward. And technically, it's doable, isn't it, Keith? I think.
Yeah, I mean, if you know, you can always store the data if it's available, and you know, and organize it, you know, in a way against locus. So technically, there's no reason it can't be done. I mean, I think it'd be quite difficult, but you know, the data structures can handle it.
Start somewhere.
I'm going to make a comment. I don't think the data are there. So you can't do any of this without there being data, and transcriptome data is extremely touchy, and coming from clinical samples is going to be even more touchy. So I think it's a wonderful idea, Bill, but I think the practicality in, at least now, is very low. I will make a comment though, and I think you're all aware of this, is that one of the issues with the data is the sampling, is that clinical labs report from one isolate per patient, and we know there are a lot of mixed infections. And there's nothing you can do on your end because it's the data that you're getting. But I still think that is one barrier to really understanding the epidemiology if you don't have the full picture of what is in the population. I don't know if anybody wants to comment on that.
Well, we've had a bit of conversation in the chat, but that's good. It seems that Helen, you've been
Linda been there.
Yeah, I mean, obviously, always adding more loci is great, but it must be quite difficult, Linda, getting the loci that you get. I mean, you know.
Yeah. So exactly. So the hi Helen, by the way, how are you? The allelic profiles 131 and 111, they are most common almost everywhere. So they are really like spread globally for some reason. And when we were analyzing also whole genomes, I mean complete genomes, like we really like covered 100% of genetic information that we got directly from clinical samples. And then we of course translated it to the MLST allelic profiles, of course. And then we could really see that they differ only in few mutations. And these differences were all the time in different positions, most of the time also in non-coding positions. So they were really like a random mutations. So I don't think it's even possible to actually find some genes that would be discriminating these two allelic profiles.
No, I know. Sorry, that's too bad. I like the scheme better than that. I'm using it almost every day.
Okay, I'm happy. Cool.
Oh, that's great. Well, it's quite interesting that even sometimes with the power of genomics, the information just isn't there. I mean, and isn't it such a tragedy that I mean, one of the good things perhaps maybe to come out of this COVID, quite apart from the publicity forum events, of course, which maybe not be seen as a universal present, but one of the things maybe that will come out of this COVID pandemic is a little bit more of an emphasis on infectious disease, because although obviously we've had this terrible infectious disease problem, there are many infectious disease problems, and I think treponema pallidum is one of them that really could be dealt with. I mean, you know, it's a sort of scandal to humanity that there's still so much treponema pallidum disease around, isn't it, really? Because it is tractable, it is soluble, and it could have been solved a long time ago if people had made a real effort. So let's hope that the post-pandemic world, we try and get to some of these infectious diseases that and really start combating, because of course it is an equality issue as well, isn't it? I know they've got the G7 happening. There's a lot of this is affecting people low income and marginalized communities and so on.
Yeah, absolutely.
Yvonne, you just one thing. Sorry, you've turned off again. But one thing I wanted to ask you is, because you obviously work in a clinical environment, do you think having some ability to link specimens that were isolated from different species from more than the same patient would be doable or useful? Or?
Yeah, I think it would be doable, and I think it would be also useful. And I actually think that some of these patients are not only diagnosed, for example, only chlamydia, but I think they also run directly the test for gonorrhea. So these data should be somewhere, and these samples should be somewhere. I think in Amsterdam it would be very, very good possible to get this kind of samples for analysis.
So I think we've got a number of jobs that have been given to us as a result of this. I think one would be to look at that, and I think that would be something we could easily think about, how we link samples across databases to enable reference labs to do that in a way that's anonymized. So you obviously don't know, you can't identify the patient, but you know that it's the same individual. And the other thing, the comment about the lineage structures and so on, I think that's again something that we need to think about.
Something we were planning on doing, that I think it would be great to have a number of people who are, experts in this, and us kind of devising something together. Definitely.
Yeah.
For the anyway, in the lineages.
Yeah, and I'm also very enthusiastic about setting up schemes for vaccination candidates, so that it would be very easy. I know that in the chlamydia field they're working on a vaccine, and it will just take a couple of years before it's ready. And then it would be great that if we have these schemes already there, or if we can adjust them when other antigens are tested or whatever.
No. Definitely possible.
Yeah, well, Charlene in our lab, as I say, who's on maternity leave at the moment, she and I know there was Ali's on the line, I think, but we were able to link that with phenotypic info from the MATS assay and so on, and to enable one to get some sort of idea. And then again, that links again back to a little bit of what Bill was saying. We're also looking at regions that predict expression. So there's quite a lot that can be done, I think, from this information. And I think and to set it up proactively, I mean, again, isn't that a lesson? Being proactive in infectious disease is a very, very good idea.
Right. Yes.
You just need somebody to be interested in it and wanting to curate that particular scheme, for example. If they just get in touch with us, we can help or set this up, organize it, and show people how to create it even themselves as well if they want to.
Yeah, we're very keen to have people who want to create things.
Yeah, creation is a long process. Can be quite complicated. So the more people that would like to be involved in this, the better, definitely.
And it's also best done by enthusiasts and experts in their area, which is why it has to be a decentralized process, because even one particular group of organisms, and I mean, the Neisseria we've got the most curators from, but in theory we can have an ever expanding community of curators for every aspect of this. I'm just looking at. So, I mean, we have gone a little bit beyond time, which is not necessarily a problem, because people can always leave, and I know a number of people have, but there's 34 people who have stayed with us right to the end, which is great. Thank you all very much for staying to this time. Odile, did you want to just to sort of tie up, unless anybody else has anything else to add? I think.
So I just wanted actually to say that if people have deposited data in sort of private projects in the databases, if they do need any of those data curated as well, they should contact one of us because it won't automatically get done. I just wanted to say that, because I've noticed there are some people on the call that might have data in private projects. So and they might not necessarily get curated unless you tell us. So that's it, really. So well, thank you very much to all our lovely speakers. I'm really grateful that I really appreciate you spending the time to record your talks. I know it's really time consuming, takes a lot longer than you imagine to record talks. So thank you very much. I thought they were really all excellent, and I really actually learned a lot from everything that you said. And thank you everybody for joining and for your questions. And yeah, that's it from me. I guess the next one will be in September, won't it, Martin?
Yes, and we'll be looking at the Holly Batch is going to be helping to organise that, and it's about the gonococcal genome library, 10 years of doing genomic epidemiology on Neisseria meningitis in the UK.
Lovely. So I guess we've got a few to do kind of lists. And,
a few action points, yeah.
Yes, a few action points. So if you like, you can email any one of us and we'll kind of set something up for all of those things and get those things sorted out. Lovely. Okay, well, thank you very much, everybody.
Thank you.
Thank you, everybody. Lovely to see you all. Thank you for coming.
Thank you very much.